Hey everybody, it's Dr. Eric Balcouch. We're back for another edition of Thyroid Thursday, and I got a lot of requests to do a video on the liver, on lipids, and thyroid hormone. So this is it. There's a lot to cover, so buckle up. It's going to be a bumpy ride, okay? If you need to, watch this a couple times. If you have any questions when you're done, put them under the video. Wherever you watch the video, put them in the comment section, and we'll try and get to them. All right? So, we need to get these lipids to the liver, and then we need to do something with them. So when we're talking about lipids, we're typically talking about free fatty acids, and the free fatty acids are typically packed together as something called triglycerides, and the other lipid we pretty much talk about is cholesterol, okay? So we get a percentage of these things coming into the liver, and then the liver's got to do something with them. So where do these things come from? Well, free fatty acids, they can come from our diet. So as we eat food, those fatty acids are broken down, absorbed, and then they get into the body, and then they head to the liver where we do something with them, okay? Fatty acids can come to the liver from the adipose tissue. So when the adipose tissue gets signals to release triglycerides, or it can release them into the bloodstream. Those triglycerides can go to the liver, where we can use those triglycerides many times as energy, okay? And then we can have de novo synthesis of fatty acids, and that means that the liver can actually generate fatty acids, and I'll explain why they would do that in a little bit. As far as cholesterol, where does that come from? Well, we can get some from our diet, okay? We can get some from the other cells in the body that have generated cholesterol, need to get rid of it, and they're typically packed up onto these HDL molecules and come back to the liver, and that's why people say it's good, HDL is good cholesterol because it's going back to the liver. Yeah, there's not good and bad, but that's one of the reasons why we think it's good, because it's bringing the cholesterol out of the body back to the liver. And cholesterol can also be made de novo. It means the liver can generate its own cholesterol, and we'll talk about how that happens as well. So the exit strategies for these things is we could use these free fatty acids to make energy. We call that beta oxidation. And that's really what a lot of people are trying to do when they're on a high fat, low carb diet, is use fats as a fuel, okay? If we don't need those free fatty acids for beta oxidation, then we can pack them up back into triglycerides, and we can store them in the liver in these things called lipid droplets. So that way, later on, when the liver needs some free fatty acids for energy, they're right there and available. If these guys are full and we don't need the beta oxidation, the triglyceride, those free fatty acids that were packed back up into triglycerides can be clumped onto another lipoprotein, and we get VLDL, and the VLDL can go out into the circulation. As far as the cholesterol, what do we do with that? Well, now that we've got cholesterol in the liver, either from our diet, from the other cells, or made de novo, then that cholesterol could, if we don't need it to do anything in particular, it could get packed up with the triglycerides, become VLDL, and go out into the circulation to support other tissues or get stored. We could use cholesterol maybe to support the cell membrane, make healthy plasma membranes for the hepatocytes. That would be a good thing. We could use that cholesterol to make bile acids. These are the things that help break down your fats, help kill the bacteria in your gut, help maintain the tight junctions, really important. Or we could just say we're gonna just get rid of them. We're just gonna push them into the bile, push that cholesterol into the bile and get rid of it, okay? Now, what role does thyroid hormone play in all of these steps? Well, to get these chylomicrons and LDL and HDL to dump their contents into the GI tract, we need what are called, or into the liver, we need what we call receptors for these things to bind to and get into the liver cells. It's called an LDL receptor, and the LDL receptor, there's another one there, don't worry about it, but they're controlled by, guess what? T3. So T3 upregulates those receptors, okay? So if we have a low T3 state, those receptors don't work so well. Matter of fact, if we have a low T3 state, there's an enzyme in the body, PCSK9, that'll just degrade those LDL receptors. So low T3 state in the liver, cholesterol, LDL piles up. We may even see and potentially an elevated HDL. Number two, where's number two? Number two, to get those triglycerides from your adipose tissue into the liver, you need more transport proteins. And guess what regulates those? T3, you got it. So if you have a low T3 state at the liver, you're gonna have triglycerides piling up in the bloodstream. Number three, we talked about de novo synthesis. So when your mitochondria, when there's a low T3 state inside the cell, inside the liver cells, the mitochondria don't work as efficiently. They can't burn as much glucose. And so then what happens is the glucose that was converted into something called acetyl-CoA and went into this thing called Krebs cycle that you learned about in 7th grade science class, can't go through the cycle to go through the electron transport chain and make all the ATP that you want for energy. Instead, it gets pushed back out of Krebs cycle, and it can, one of two things can happen to it. It can either be converted into more free fatty acids, or it can be converted into cholesterol, okay? And guess what you need to convert that citrate into, kind of run this HMG-CoA, which is the main enzyme that converts and produces cholesterol. You need T3, okay? So number five, where are we at, number five? Oh, we talked about fatty acids. We can burn those to make energy, called beta oxidation. But what do you need to regulate beta oxidation? You need T3. There's an enzyme that's, where is it? CPT1. This is the enzyme that kind of helps transport the free fatty acids into the inner mitochondrial membrane, is regulated by T3. So if you have a low T3 state, you may not be able to burn the fatty acids. So if you're going on a low carb, high fat diet, and then you're like, hey, I don't feel good, I don't have energy, I'm not losing weight, it's because you have a low T3 state to be able to burn those fats. T3, number six. Oh yeah, we said that if we have extra fatty acids and I can't burn them in beta oxidation, I can store them in these lipid droplets. And those lipid droplets, when we want to use the free fatty acids in those lipid droplets, we need an enzyme to kind of hydrolyze and release the free fatty acids. Guess what? That enzyme, this adipose tissue, adipose triglyceride lipase, guess what it needs? It needs T3. So if you have low T3 inside the cell, you can't break down these lipid droplets inside the cell. It's going to lead to something along the lines of fatty liver disease. Now, we've got number seven. So seven is, hey, I want to make bile acids, but guess what? You need T3. T3 regulates the primary, one of the primary bile acid genes, CYP7A1. That's the rate limiting enzyme to make bile acids. So if you have a low T3 state, you can't make sufficient bile acids. Number eight, if I just want to get rid of the cholesterol, get it into the bile, I don't even want to make it into bile acids, I just want to get out of it, get rid of it, I can push it through this transport molecule and push it right into the bile. But guess what? That ABCG5 receptor or transport protein, it requires, guess what? T3, all right? And then we talked about T3, if you have a low T3 state, this enzyme PCSK9 actually degrades the LDL receptor, and now I can't get those fatty acids into the, and cholesterol into the liver, and they stack up in the bloodstream. So what are the signs of low T3 in the liver? How about this? I'm gonna have a low T3 or free T3 in the blood. Just look for it. If it's low, it's probably a good indication that we're probably gonna have some problems with the liver. Number two, look for a low free T to free T4 ratio, meaning what this means is that you might have plenty of T4, but you're not converting it well to T3. You could even have normal T3 and normal T4, I should say free T3 and free T4, but if the ratio is low, you're probably not doing a great job converting that T4 to T3 efficiently. You could have high cholesterol and LDL in your lab work. You could have low bile production. Which means you're gonna have problems with fat and breaking down fats and digestion. That may also lead to bacterial overgrowth and leaky gut and food sensitivities. You could have high triglycerides and VLDL. You could have decreased insulin production or loss of your first phase insulin response due to changes in bile acid concentration. You could have glucose or insulin resistance, meaning both of those could be high. You can have high ferritin and you can have high or low CRP. That's it.